Welcome to Micro Seminar this early morning out here in California. I'm Lizzie Wilbanks, and I'm really happy that all of you who have either woken up early in our time zone or who are ending your day, if you're over in the European time zone, thanks for tuning in. It is my very great pleasure this morning to introduce Dr. Annie Rowe from the University of Cincinnati. She's just started there this year as an assistant professor, and if you are looking for exciting new labs doing exciting things, you should definitely go look her up. She's a very good friend of mine and a fantastic scientist. She did her graduate work at Cornell University with Ruth Richardson, looking at dihalococoides and methanogens and thinking about what sort of biosignatures for their respiration and activity look like, and then went on to work at USC on electromicrobiology, thinking about all things good about bugs and currents that they make, with Ken Nielsen, Yamam Ahmed, and Moel Najjar. And she's going to tell us a little bit about some of her postdoc work and then some stuff that's hot off the presses from her new lab. So without further ado, Annie, why don't you take it away?
All right. Thank you, Lizzie. All right, let's see if I can do this. Great. Cool. So hopefully you guys can see that.
Yep, looks great.
Great. Okay. Yeah, so thank you, Lizzie, for inviting me to do this, and thank you to all of you watching this, whether it's right now or in the future. So I'm going to tell you guys a little bit about the work I've been doing looking at microbes that can mobilize electrons from mineral phases, electrodes, and or even other microbes as a way of acquiring energy. And so we can colloquially think of this as eating rocks. And this really kind of harkens to one of my favorite things about microbiology, the fact that, even though these mobilizing of electrons or these redox processes are really central to all life, the really cool redox processes happen in the microbial world. And today I'm focusing on one that, though we've known about it for a long time, has really never been well characterized mechanistically. And so I can tell you a little bit about how I've been investigating that. So my interest in this area very much started with observations of organisms breathing rocks. And so at this point, you've probably heard about organisms like Shewanella that has the ability to respire these solid phases. So this is a manganese oxide particle, and hopefully if I can get back to something that will allow me to point. You can see this video here of Shewanella swimming around and actually reducing this manganese oxide particle to soluble Mn2. And this metabolism, I think, kind of turned a lot of the paradigms of respiration and metabolism on its head when it was first discovered. Because if you think about it, how do organisms interact metabolically with this, insoluble amorphous material that's often thousands of times bigger than the cell? And we now know that mechanistically this happens through Shewanella transporting electrons from sort of the inside of the cell through its protected membranes to the exterior of the cell. And because of that, it's not so important what sort of sink this organism can use, and we can actually substitute these mineral phases for things like this clear conductive material, this ITO electrode that we can poise at just an energy that's favorable for accepting electrons. And in the process, we can see, again, this is a video of Shewanella growing and attaching to these electrodes, and we can actually measure current in the process and do a wide variety of electrochemical tests. And so using a variety of techniques, both basic microbiology and combining electromicrobiology techniques, we've been able to pretty well characterize how organisms like Shewanella do extracellular electron transport. So in that video of Shewanella swimming around and interfacing with the mineral and or electrode, there's a direct interaction that's mediated through a series of proteins that these organisms have that are multiheme cytochromes. So there's a series here of redox cofactors that allow electron transport from the inner membranes of the cell to the exterior of the cell. And even in a non-modal phase, so when Shewanella, lays down and starts to form a biofilm, it can actually still utilize this mechanism. In this case, it can produce appendages. In Shewanella, we know these are extensions of the outer membrane that also have multiheme cytochromes, but that in this case can allow lateral electron flow across the length of these nanowires or appendages. And in theory, these appendages can interface with some sort of redox active substrate and/or other cells. And the final mechanism is the ability of organisms like Shewanella to interact with redox active mediators, so soluble compounds. In the case of Shewanella, Shewanella produces and excretes flavins that it can reduce in the extracellular environment, and then those reduced compounds can diffuse away and interact with some sort of other solid redox active surface. And so this is potentially one of the more ecologically complex mechanisms. And in fact, it's been shown that there's some interaction between these two. But when it comes to thinking about eating rocks and mechanistically what's going on here, we really don't have a good understanding of what the mechanisms might be. So especially in the extracellular electron transport world. So there's potential that really we're just looking for what are reversals of the same types of mechanisms, whether it's these same multiheme cytochromes or similar appendages or redox active mediators. But again, we don't have specific detail about how these processes might happen. And this is somewhat surprising because this is not a new metabolism. So we've had observations of organisms that can metabolize inorganic materials for a long time. So this is one of my favorite examples of Vignettes, which form these little, insoluble sulfur granules within the cells as a means of energy storage. And so it can store this sulfur for respiratory purposes and incorporate it into its metabolism. And this is something that, was seen by Wynogradsky back in the late 1800s. But we don't still yet have a good idea of how this organism and/or really any other organisms are utilizing things like elemental sulfur in their respiration. And part of this has to do with the fact that it's been really challenging to culture a lot of lithotrophic organisms under laboratory conditions. And Vignettes is a good example. So even though we've known about this organism for over 100 years, we still don't have robust cultures of this organism in the lab. So using genomics, though, we can see that there is some evidence for the capacity of lithotrophs, things like iron oxidizers, like Siderioxidans lithotrophicus, or even Pseudomonas palustris, a phototrophic iron oxidizer, to perform or at least have some of the genes involved in extracellular electron transport. So this is homology between Shewanella onidensis and these multiheme cytochromes that we can see in some of these iron oxidizers. But there are some gaps in this information, and it's not clear exactly how these proteins might incorporate into processes like iron oxidation. So there's still a lot to learn here. And from an environmental perspective, I think it's important to try to understand how these processes work, because on the one hand, it's challenging just from geochemistry alone to detect lithotrophy in a lot of environmental settings. So a lot of the intermediates or the products of these metabolisms are very transient, very reactive, and often don't have long half-lives in the environment. And so geochemically, they're challenging to detect. And if we look at the traditional techniques we utilize as environmental microbiologists, where we investigate nucleic acids, we look at either taxonomy or functional genes, this approach kind of breaks down if we don't have good information on what mechanisms, what genes are involved, or even what taxa are capable of performing some of these processes. And then from an applied perspective, these organisms that have been able to interface with solid phase materials, things like electrodes, have opened up a wide array of applications. And so just looking at organisms that breathe electrodes, there's a lot of potential for wastewater treatment strategies, different, treatment of ammonia or organic compounds, in the process of generating a little bit of energy. But I think what's really exciting is that on the cathode end, there are also a whole slew of applications, including,
Annie,
yeah?
I'm sorry to interrupt you. There's a little tiny banner that's up on your screen that says Google Hangouts is sharing. Can you see that?
I can. Do you want me to hide it?
Yeah, because it's actually running in front of some of your really awesome graphics. Okay, thank you.
Yeah, no problem. Sorry, I didn't realize everybody could see that.
No worries.
Okay, so on the cathode end, we also have a whole slew of applications of organisms that are able to take up electrical energy from these electrodes and convert it to different substrates. So things like potential biofuels, processes that we could utilize for remediation of things like heavy metals. And I think it's important to realize that the mechanisms here are really pretty poorly understood, especially for these direct interactions that organisms might be having with things like cathodes. And so I've been delving into this area in a few different ways. And as Lizzie said, it's sort of a mix of some of my postdoc projects and things that I'm currently working on. But I'm going to tell you just sort of 3 little short stories about ways I've been looking at electron uptake and/or organisms eating rocks. And the first one is going to have to do with our model system, Shewanella, a project I call Shewanella on life support, where we really looked at the ability of this organism to conserve energy from electron uptake. So I introduced Shewanella, of course, as a rock-breathing organism. So you might be curious as to why I'm now talking about electron uptake. But one of the things I didn't mention was that this mechanism of extracellular electron transport in Shewanella is actually a completely reversible process. So all of these heme cofactors here have very similar redox potentials. And what that means is depending on the potential you poise on an electrode or the redox potential of your surface that these organisms are interacting with, electrons can actually flow in either direction. And it's been observed that we can actually see electron flow into Shewanella cells on things like cathodes. But I think one of the questions is this actually a physiologically relevant process? So what happens to this electron once it gets past this sort of reversible process? Does it interface with anything in the cell that would allow energy conservation? And so that's one of the big questions I wanted to look at. And so in order to do that, this is just a blow up of the inner membrane of Shewanella, where energy conservation would happen. And this organism has both normal pathways for anaerobic respiration and aerobic respiration. So it has all of the proteins that one might need for doing oxygen reduction or things like denitrification. And our question is here, how are the electrons entering into the cell and/or are they entering into the cell in such a way that would allow them to interact with, for example, these terminal cytochrome oxidases? And to look at that, I utilized some microscopy along with electrochemistry, and in particular, the use of this redox sensing dye that interacts with high potential cytochromes and is reduced by these high potential cytochromes and fluoresces green. And I coupled that to use of electron transport chain inhibitors, things like antimyosin, which is a ubiquinone mimic, and/or cyanide, which is a high-spin cytochrome inhibitor. And what we can see, this is a movie that's basically a biofilm. Hopefully I can play the video. So this is a biofilm. You can hopefully make out the cells here in red. So this is a lipid stain. And as soon as we poise a potential on this electrode, so this is a clear conductive ITO electrode, as soon as we poise a potential, we can see an accumulation of this green color. And this is a lipid-soluble molecule. So if we turn off the electrode and/or we treat the cells with an inhibitor, something like cyanide, we see that eventually this green signal and this accumulation starts to dissipate. And we see a lot more of that red lipid dye or lipid signal here. And this corresponds really nicely to what we see in terms of current. So we see a good amount of electron uptake. So this is electron consumption or cathodic current here. And as soon as we treat the cells with something like antimycin or cyanide, we see a dramatic sort of increase in current, I guess, or sort of a drop off in this current consumption. And this is really nice because, you know, inhibiting things like ubiquinone and or some of these terminal oxidases suggests that potentially we have some proton translocation at these points, which would allow, in theory, these organisms to make an electrochemical gradient from this electron flow. And so to better support that, we also quantified ATP levels in the biofilm. And so this is just a comparison of the ATP ratio of ATP to ATP and ADP under these cathodic biofilm conditions and or a condition that was treated with an uncoupler, so something that gets rid of this electrochemical gradient. And it was treated with an uncoupler for about 3 hours. And so we can see a nice significant difference in ATP levels, which I think helps support that these cathodic processes are involved in energy conservation. And so in addition to that, though, you know, we also wanted to look at whether or not, in addition to this forward reaction where electrons can flow in a downhill reaction to oxygen, we could potentially get electrons into the reducing equivalents of the cell, which is potentially a more slightly uphill reaction. And this is something that's been postulated in a lot of Chemolithoautotrophs. And so it's something we wanted to investigate here. And we utilized luciferase as a way of sort of monitoring or assessing the redox pool in the cell. So here, this is a luciferase enzyme that utilizes FMN, a little bit of oxygen and a fatty acid to produce light. And this FMN can be regenerated by NADH, and NADPH is often involved in restoring fatty acid, the fatty acids here. So there's a lot of places where reducing equivalents entered into this equation. And so utilizing this as sort of an in vivo marker, we wanted to test the potential for reverse electron flow. So thinking about how electrons would make their way into the reducing pool, it had been postulated that the sort of electrochemical gradient that you generate by this forward reaction, so coupling electron flow to oxygen reduction, in theory, when you sort of oxidize or highly oxidize your NADH pool, could help drive reduction, so the reverse process of this NUO or this complex one. It could help drive that process. So essentially, utilizing this electrochemical gradient to drive electrons in the reverse direction. And so to test that, using this in vivo marker, we also coupled or utilized an uncoupler here to try to link what was going on in the redox pool with this electrochemical gradient. And what we found is, so this is examples of light production in a few wild type MR1 biofilms. As soon as we treat these biofilms with an uncoupler, we see a dramatic decrease in light production. And so this suggests that we have a link between what's going on in the redox pool and that electrochemical gradient that we're seeing under these cathodic conditions. So one might argue that perhaps this is an effect that is linked still to carbohydrate metabolism. Well, it turns out if we add a carbohydrate, here we're adding lactate, we actually can inhibit this effect. So if we add lactate to these biofilms, we see that addition of CCCP or our uncoupler prevents actually seeing that decrease in light production compared to wild type conditions. And sort of as a third line of evidence, we also deleted the complex one, so that gene or that enzyme complex that is responsible for the reversible reduction of NADH from electrochemical gradient. And I should note that Shewanella does have multiple quinone oxidases, but this is sort of the main one that's been linked to proton pumping, and in theory would be important for reverse electron flow. So if we delete that, we see that we no longer see that sharp drop off when we add iron coupler. So in fact, we see almost a little bit of stimulation in light production, and it takes a good 5 to 10 hours for light production to drop off under these conditions. And so this starts to support that under cathodic conditions, some of these electrons, and coupled to this generation of an electrochemical gradient, in theory can make their way into the reducing pool. So it requires proton motive force, it requires an oxidized NADH pool, and it also requires this reversible complex 1. So just to sort of summarize, we have some pretty good evidence now that these organisms are able to make an electrochemical gradient, they can make ATP, they can make reducing equivalents, but they should not, in theory, be able to grow via this process. So they don't have, Shewanella does not have a means of carbon fixation, and so it's still going to need a source of carbon for growth. And when we tried to look at, cell numbers over these cathodic experiments, we never actually saw any growth, but what was interesting is we also didn't see significant cell death. And in all the other control conditions we looked at, whether or not it was a poised potential but an anodic current, or even just open circuit controls or planktonic cells, in all of those conditions over the time frame of these experiments, we were able to quantify significant cell death or cell decay. And again, we didn't see that under these cathode conditions. And so this helps support that there is some energy generation going on in these systems. And I think this is kind of exciting from a linking electromicrobiology with systems biology perspective, because it allows us to think about funneling electrons into the cell in a variety of different ways that will allow us to maximize things like a forward process like heavy metal reduction, denitrification, and/or any sort of specific process on this end. But it also gives us the ability to perform some biosynthesis. So being able to get electrons into the reducing pool potentially allows us to do things like generate, some sort of biofuel using these processes. And again, this is going to sort of maximize the amount of electrons that would be devoted to either heavy metal production or biofuel production, as opposed to growth, because this is not, again, an autotroph. So the last thing, most of this focused on energy. We did do some work with the mutants that we have in Shewanella, trying to look at exactly how this electron uptake process happens. And the short story is that it's a little more complicated than just a reversal of the known extracellular electron transport pathways. And so to try to get at that more sort of broadly and/or sort of from a bigger perspective, I've been collaborating with Buzz Barstow at Cornell, who has an entire genome knockout collection in Shewanella. And he's been looking at trying to investigate electron uptake via these mediator processes. And he's highlighted a slew of mutants that he thinks are interesting. They're not just our characteristic extracellular electron uptake organism bugs. And we've been screening them for a phenotype on these cathodes. And this is work that's done by my soon-to-be graduate student, Leah Trutschel. But, so moving on. So Shewanella is a cool story, and it's a great model system. But even though it can make energy from this process, it's not likely to be evolutionarily adapted for electron uptake. And the truth is there are probably a wide variety of organisms in the environment that are. And so also as part of my postdoc in Ken Nielsen's lab, I went out to the environment and began to look at trying to assess organisms that are going to be better model systems for rock-eating microbes. And so as I said, so the big questions here are there organisms that are better adapted to solid substrate oxidation and sediments? And if there are, how are they accomplishing their extracellular electron transport, or how are they accomplishing these mineral oxidation processes? And so this work was done at Catalina Harbor. Catalina is an island that's about 25 miles off the coast of Los Angeles. Catalina Harbor is a Pacific-facing harbor, and we chose this area in part because we have access to a research station there, but also because it's a sort of high mineral content sediment. It's got a lot of iron, and it doesn't have a lot of carbon inputs. So there's not a lot of people living in this area. It's a relatively unpopulated region. And so it seemed like a good environment to go and look for sort of lithotrophic metabolisms in a marine setting. So we collected sediment from this environment. We utilized some pretty fancy electrochemical techniques. So this is a 3-electrode system where we can poise potential on a working electrode. And here we're poising it to allow electrons to be taken up, hopefully by biology in this system. The potential is being poised relative to a reference electrode. So this is a silver-silver chloride reference. And there's also a counter electrode in the system that will make up or keep electron balance here. And utilizing this enrichment system, we saw good evidence of biological electron uptake over time. But what we really wanted to do was sort of isolate some of these organisms. And so we took these enriched electrodes back to the lab. We looked at coupling this process with the terminal electron acceptor. So now we've taken the sediment away and shown that, for example, if we add something like nitrate, a potential terminal electron acceptor, we can couple very nicely electron uptake. And just as a note, this is negative current here. I've inverted the axis. So we were able to see, again, both in the sediment and the lab, good evidence for biological electron uptake. We then isolated organisms or further enriched organisms on solid substrates and ultimately isolated them using a variety of sort of solid media isolation techniques. And we isolated we isolated a lot of different organisms. And I think, to try to winnow down which were going to be sort of the best organisms to start studying, I took my cue from Shewanella, which has really, I think, been a powerful model system, in part because it is a facultative heterotroph. And so a lot of these organisms that I'm showing here are actually facultative lithotrophs. So they have the ability to grow lithotrophically using iron or sulfur minerals predominantly, but they also have the ability to grow on an enriched media in oxygen. And so I've been looking at a variety of these to try to better understand sort of the basics, or the low-hanging fruit, I guess, of electron uptake in these organisms. And so this is just an example of a project I worked on with an undergrad in Ken Nielsen's lab. So she helped me characterize this Thioclava electrotrophica organism, which again is a heterotroph, but also capable of autotrophic growth and of electrotrophic growth. And so we've been able to, now that we have these isolates, do some sort of further electrochemical characterization to try to compare across strains. And so here again is our 3-electrode system that we're utilizing in the laboratory. And we can break apart the system once we get nice evidence of electron uptake. We can look at electron uptake potential in biofilms under the planktonic cells and the spent media. And we can also do tests like cyclic voltammetry, which is sweeping voltage over time that allows us to compare relationships between current and voltage. And it gives us a sense of what the features are like that are involved in electron uptake. And in particular, what redox potential or what energy level do we see electron uptake happen in these different strains. And so this is some work that's been done by a graduate student in Mo El-Najjar's lab, Amruta. So she's been looking at denitrification, so an anaerobic cathode system with Thioclava, that same organism that Rachel has been characterizing. And she showed addition of cells in the presence of nitrate gives us some nice electron uptake signal. This is biologically mediated. So if we hit it with something like azide, we can really mitigate this effect. And she's also done a lot of work showing that this is a hydrogen or hydrogenase independent effect. And so we can do things like do these CVs in the presence of a lot of hydrogen, and we really don't see any difference in terms of the electron uptake potential here. So this is, a close-up of one organism, but if we start to compare across a variety of different organisms and even compare it to our organism like Shewanella, we see that all of the isolates have a slightly different redox potential where they start to take up electrons. And if we, all of these experiments here are normalized to electrode area. So we can see that even using the same, flat surface electrode with the same area, there are differences in the capacity of these organisms to take up electrons. And in some cases, you have almost 5 times more electron uptake capacity in some of these organisms. So from here, we wanted to see, kind of investigate what are the genomic differences that underlie some of what we're seeing in their electrochemistry. And this is work I actually am collaborating with our illustrious host, Lizzie Wilbanks. Unfortunately, that slide got cut out. But so Lizzie helped me put together some nice draft genomes of these organisms. And of course, the first thing we did was try to look for homology to known extracellular electron transport genes. And good or bad, we didn't really see very much homology. I think our only hit was this octaheme cytochrome in a Marinobacter that really only has 30% known identity to any other protein. So it maybe kind of looks like a multiheme cytochrome, but that's really where the homology ends. And so we kind of had to take a coarser view of what's going on with the genomes of these bugs. And so we started by looking at just the cytochrome-containing genes, in part because it's been shown that electrochemically active organisms tend to be enriched in these C-type cytochromes. And we found that that was true in our genomes as well. So this is a cluster of genomes in this Pseudomonas organism. And we can see that there are upwards of almost 70 cytochromes in this particular organism. And this is true for all the different isolates that we saw. So these are different strains within sort of a genome cluster here. And all of them have at least equal to, if not quite a bit more cytochromes than organisms like Shewanella. So this is a lot. So it's hard to know what to do with this information. So what we're looking at now, or what I'm looking at now, is trying to utilize high throughput molecular studies to try to understand what genes are going to be involved in electrotrophic growth versus something like heterotrophic growth. And so this is hopefully going to be utilizing some techniques like TN-seq. And right now, I have 2 undergraduates in the lab who are sort of helping me in this area. So one is looking at developing a genetic system in organisms like that Thioclava. So he's been able to get a plasmid into Thioclava. And then together, we're also doing some long-read sequencing, also collaborating with Lizzie Wilbanks and trying to get sort of complete genomes for doing sort of more molecular characterization here. And then in addition to Thioclava, I have another undergrad who's looking at an Idiomarina strain. So one of our other interesting bugs that has really a pretty small genome and a pretty interesting electron uptake phenotype and makes really crazy cool biofilms. So hopefully we'll be able to tell you more about that later. But just to summarize, I think what I found really impressive about this project is that this is one sediment, and a sediment that we incubated at 1 redox potential. And we observed a whole new slew of organisms that were capable of cathode oxidation, or these electron uptake. And I think this speaks to an immense biological diversity of these processes. And this is, further supported by looking at their genomes and seeing that, they have a whole slew of these cytochromes that are unique and distinct, even across each other. So there wasn't a lot of homology between the cytochromes, even within these different groups. And so I think hopefully we'll be able to tell you a little bit more about what some of these cytochromes are doing and what they're involved in terms of electron uptake. Okay, so the last little story, and the one that's sort of hot off the press as well, it's not even in the presses yet, so it's one we're still working on a publication for. But I've also been looking at electron uptake in methanogenic organisms. And here, the electron uptake, rather than interactions with a mineral, even though mechanistically it's likely very similar, is probably more of an interaction between different microbial members. So things that would happen in syntrophic communities. So I think what really brought me to this area, or thinking about using organisms like methanogens on electrodes, has to do with this overall sort of logic for electrosynthesis, which is the ability to convert electrical energy, most likely from some sort of solar or wind processes, to some sort of biofuel or liquid fuel that we have infrastructure for already, and or as a means of storing energy from these alternative energy processes. And one thing that I've alluded to a little bit in the Shewanella world is that the majority of electrons entering into the cell are going to be coupled to the respiratory process, so coupled to this downhill reaction to a terminal electron acceptor. And if we're thinking about making biofuels, we're going to have sort of a limited efficiency here, just based on the fact that the majority of our electrons are going to be going towards energy. So can we get around that by utilizing an organism that makes a biofuel as its main respiratory process? And so this is something I think is really pretty exciting about using methanogens in these electrochemical systems. And so I'm definitely not the first person who's thought of this, but as of now, really the only mechanism we have for organisms that do this is one that's been shown by Jörg Dutzmann and Alfred Sporman at Stanford, where they looked at the ability of extracellular enzymes to actually attach to carbon electrodes and mediate electron uptake and form things like formate or hydrogen in these electrochemical systems that then are metabolized by methanogens. And so this is sort of thought to be the main mechanism for electron uptake in these electrochemical systems for methanogens. But what's interesting is this work was done in a methanogen that does not contain cytochromes. So there are these two major lineages that within the methanogens, ones that contain cytochromes and ones that do not. And there are some very important biochemical distinctions here. And if we look at methanogens with cytochromes, there actually is some evidence for either a hydrogen-independent electron transfer or sort of even a formate-independent electron transfer, based on some evidence for interactions with Geobacter. So this is work that was done by Amelia Rowe, looking at a co-culture of Geobacter with Methanosarcina barkeri, which acts very similar to a syntrophic fermentation. So you can couple ethanol oxidation in the presence of both of these organisms and see enhanced methane production that you don't see when these organisms are grown alone. But interestingly, Geobacter does not have the ability to make hydrogen and or formate, so other substrates that Methanosarcina barkeri might use. And so it's been postulated that this is direct electron transfer between these two organisms. And so utilizing this methanogen with cytochromes, I wanted to look at whether or not we see evidence in an electrochemical system for this sort of direct electron uptake. And at the same time, I wanted to make sure we're accounting for these spent media effects of these hydrogenases attaching to electrodes. And so this is a basic experiment done in Methanosarcina barkeri cells where we have the growth media or the spent media, and one where we wash away the spent media. And we can see that in both cases, we see electron uptake occurring, albeit it's quite a bit lower in the cell-only experiments. But it does suggest that there's still some evidence of electron uptake when we get rid of, the potential contamination of extracellular enzymes. And we wanted to make sure that this is the case, or that we were looking at, sort of distinct mechanisms. And so to investigate the mechanisms, we again used our cyclic voltammetry. So this is our voltage sweep technique, where we can compare current versus voltage. And we can look at things like we saw in our cathode oxidizing organisms, like the redox potential, where we start to see electron uptake happen. But there's sort of a variety of features we might expect here, including processes that are reversible, something like a hydrogenase that can be involved both in reduction and oxidation. So this is sort of just examples of two of the things we might expect to find. And when we looked at, again, cyclic voltammetry plots of these wash cells versus the cells with their spent growth media, what we saw is that in CVs with both the spent media and the cells, we actually saw multiple features here in the CV. And so you can kind of see one here right around this -400 region, and then also sort of a lower potential wave going on here. And this is something we also see in the wash cell only. And it's much stronger in the wash cell only compared to the one that has both the spent media. And this is just sort of focusing in on these CVs. So this is a first derivative plot just to try to give us sort of a location of that peak feature here. And what we see is that in, again, our wash cell only, we have a pretty dominant feature out here in sort of the -500 range. But that we see some other features when we have the spent media around. And to investigate whether or not one or both of these features are involved in hydrogen uptake, we utilized the reversible nature of hydrogenases. And so we added hydrogen to these reactors and saw that one of these features in the spent media, in the presence of hydrogen, produced this really nice reductive wave. So this is really nice evidence that this is actually a hydrogenase that now is capable of hydrogen reduction under these conditions. In the wash cell only experiments, we didn't see great evidence for the hydrogenase, but what was sort of more exciting was that second feature did not seem to be affected by the presence of hydrogen. So this seemed to have sort of a hydrogen independent phenotype. And so we can compare these two again on our first derivative plots and see this sort of nice hydrogenase feature compared to one that appears to be hydrogen independent. So this work was done in wild type cells. And so to sort of further confirm that these features are not involved in hydrogen and also involved in methane production, we utilized a hydrogenase deletion mutant that was made by Gargi Kulkarni and Bill Metcalf's lab that is still capable of electron uptake in these cathodic systems. And so this is just two examples of different redox potentials where we can see electron uptake in this organism compared to spent media. If we add hydrogen to the reactor, we see that there's no effect on current in either the spent media or the wash cells. And so it doesn't seem to be inhibited in any way by the presence of hydrogen. So this sort of suggests that there's not a significant amount of hydrogen even being produced in this system. And then if we add in an inhibitor of methanogenesis, we see that in the presence of cells, this almost entirely knocks out our current consumption. And so this is pretty good evidence that not only is this electron uptake hydrogen independent, but it's also linked in some way to methanogenesis. And we can see that again with our CV plots. So we see a very similar feature to what we saw in the wild type in our wash cell experiments. This feature, again, is not affected by hydrogen. So if we add hydrogen, we don't see really any difference in that electron uptake feature. But we can inhibit it by utilizing something like BES. So this is kind of nice evidence for direct electron uptake in methanogens. It doesn't really tell us what that feature is, but I think it does speak to a lower potential electron uptake feature in methanogens. And I think that kind of starts to get at maybe what an energetic trade-off might be in these organisms for these direct electron transfer syntrophies versus sort of the traditional hydrogen intermediate or formate intermediate. And so I'm hoping to investigate this more in my new lab. And in part, I'm gonna hopefully be looking at some model communities or model coal cultures with centrifugal fermenters like Geobacter, and beginning to look at what sort of genes are involved in both of these processes, and hopefully try to tease out what type of feature might be responsible for this effect. But yeah, there's lots to do there. But just to sort of emphasize, I've shown you 3 stories, and I think what's exciting for me is I think microbes are really giving us new insight into electron transfer reactions. So I've told you a few stories about an organism that's normally a mineral reducing bacteria that can also take up electrons as a way of generating some energy. I've told you about some organisms that are mineral oxidizing organisms, and also organisms that can transfer electrons, likely via very similar processes between each other. And yeah, there's a lot more probably to do in this field. So I'm kind of excited to be here and what hopefully we're gonna find out. And just to acknowledge, so Ken Nielsen was my first postdoc advisor. I also was fortunate enough to work with Mo El-Najjar and Yana Men at USC. So a lot of the methanogen work was done in their lab. I also worked with Akihiro Okamoto, who was formerly at the University of Tokyo, where I worked with him at a JSPS short-term postdoc. And then I have also some wonderful collaborators at the University of Minnesota, Jeff Grelnick and Lizzie Wilbanks at UCSB. And here are some lovely photos of the people, with the exception of the Nielsen lab, because we were never organized enough to take a photo. But now I have my own little lab group. So hopefully we'll have some good follow-up work in the future. And with that, I'll take any questions, if anybody has any. Ooh. Ooh.
Hey, Annie, thanks so much. That was awesome. All right, so now's the process where if you have questions and you're watching, tweet us your questions at MicroSeminar on Twitter, or if you're here in the Hangout with us, feel free to chime in. That was awesome, Annie. Thanks so much. It was really awesome to catch up with what you've been doing. While folks are organizing themselves and getting their Twitter bots loaded, I thought I'd lead off with a question. So I was really excited about your direct electron transfer stuff in the methanogens. What do you think are, it's kind of basic, but what's your gut instinct for what some of the challenges that a methanogen would have that might be different from something that your other lithotrophs might have or that Shewanella might experience? What do you think would be some of the things that might either make that really efficient or really hard for them?
Yeah, well, so I think one of the things that I think has been challenging about working with this system in cathodes, and one of the things we had to be really careful of, is that the feature that we're looking at is actually lower in energy than a hydrogenase, right?
Oh, stop sharing your screen so we can see you.
Oh, sorry. I can see you.
That was very meta.
No, it's okay.
Okay. All right, sorry for interrupting. Go ahead.
No, okay. Can you guys see me now, or is it? Okay. All right. Yeah, so I think one of the things that's interesting is that electron uptake feature, I think it's lower in energy than a hydrogenase. And so one of the challenges with methanogenesis, the way that biochemically it's done, is that the first step in methanogenesis requires ferredoxin. And ferredoxin is actually lower in energy than hydrogen. And so one of the, sort of biochemical challenges and one of like sort of the big questions in methanogen biochemistry is how do these organisms make ferredoxin from hydrogen? And I think one of the energetic trade-offs with being able to do direct electron transfer and being able to like transfer a lower potential electron is that maybe you can get around sort of that first energetic back step of having to make ferredoxin from hydrogen. And I didn't talk about this just because I tend to confuse people very much with the biochemistry, but it's kind of interesting. The syntrophs themselves actually generally make, when they're making oxidizing ethyl, actually make ferredoxin. And so there's kind of this weird step where they make ferredoxin, they use that ferredoxin to make hydrogen, which is an energetically favorable process. But then now you've got these methanogens that have to take that hydrogen and do a little bit of a, some biochemical magic to try to make ferredoxin again to get that first step in methanogenesis going. So I think that's kind of the potential trade-off. The drawback, right, is that you're hitching yourself to one organism instead of getting electrons from a whole bunch of other things. So you got to be certain that that's a pretty, I think, common ecologic trade-off in organisms. But
there's no such thing as a free lunch.
Yeah, no. Yeah.
Awesome. All right, does anyone else in the Hangout have some questions before we check Twitter here?
I want to follow up on that a little bit. I understand, like, the challenge of reverse electron transport or something like this, where like an electron donor couple comes in at a redox potential lower than NADH, and so you have to push electrons back up the tower, so to speak, in order to fix carbon. Mm-hmm. But what is it about why is it energetically, what is, maybe I'm misunderstanding it, but it seems like the way I interpreted that was that you said that the actual production of ferredoxin is at a lower redox potential than the hydrogenases?
Right. So, I mean, just to put general numbers on it, let's say hydrogen's around the negative 400 millivolt window. Ferredoxin's probably closer to negative 500.
For the couple.
Yeah. So if you're talking about who would get the electrons, right, for the couple,
yeah.
Ferredoxin being the more reduced, right, is energetically favorable to give electrons to the hydrogen.
Yeah.
But that reverse process is not energetically favorable under most physiologic conditions. And so methanogens have two tricks. So one is that the organisms that I've been working with have these energy conserving hydrogenases that utilize an electrochemical gradient. So these are actually, I believe they're homologous to the complex ones, actually. But basically they utilized, a sodium gradient to basically drive electrons from hydrogen onto ferredoxin for that first step. The other lineage of methanogens actually utilizes this electron bifurcation scheme, where basically they have this last really energetically favorable step of methanogenesis, right, where you make methane. And they use a flavin to couple that really energetically favorable last step with the first not energetically favorable step of making ferredoxin. And so...
Okay. I think I misinterpreted. Well, I thought you were talking about the actual production of ferredoxin as opposed to just the, like, reduction oxidation reactions of ferredoxin.
Oh, yeah, no, sorry. Yeah, not the biosynthesis of ferredoxin.
Okay.
Right. No, sorry.
I might have just misheard you.
No, I probably am using the wrong terms. Yeah.
It's okay.
It's just the production of ferredoxin, or producing or generating reduced ferredoxin.
Generating reduced ferredoxin. Okay. Okay. Yeah. Well, in any case, it's a good review of internal methanogen cellular metabolism. This is good to have on the
Doesn't
record.
everybody just love that? I mean, come on.
I can just turn this on for my classes rather than messing it up.
Totally.
All right, Annie. So we've got a question here from Tyler Arbor, who is joining us from the University of Ghent.
Okay. Hi, Tyler.
And Tyler asks, oh, damn it. Where did I put it? I cannot use Twitter. Okay. Tyler says, for the last part with methanogen hydrogen versus direct electron uptake, do you think you can rule out significant hydrogen production at your cathode so that adding more won't matter, especially given the low solubility of hydrogen?
So that adding more hydrogen won't matter, or?
I think so, yeah.
Okay. Yeah, so I mean, the hydrogenase deletion mutant, we could actually do those experiments in an 80% hydrogen headspace, which gets you pretty close to a millimolar concentration of dissolved hydrogen. And we didn't really see an effect on the cathodes in terms of current consumption. And we did see that in the wild type, and in particular the wild type with the spent media. And in fact, so when we added 80% hydrogen, we were actually able to switch the direction the current was going, I think because we had these hydrogenases that now could energetically take up hydrogen from the system. So, I mean, it is a bit, I would say that in the cathode world, especially utilizing these really reduced electrodes, we have to be careful about the hydrogen issue. And one of the things that I tried to do was I'm working really as close to that, zero redox potential of a hydrogenase as I can, in part to sort of, because a lot of the other electrochemistry people use, several hundred millivolt more reduced electrodes just because you get such stronger current and more biocatalysis. But from trying to understand what's going on with the microbiology perspective, working kind of at these lower current regimes, but ones where it's a little easier to kind of tease apart what's going on, I think was kind of beneficial.
And just to clarify, that was essentially the question in your answer was talking about abiotic signal that you might be getting from hydrogen.
Right. Yeah. Yeah. And so without hydrogenases there, we don't really see. So hydrogen on these sort of electrodes takes quite a bit of overpotential to actually get significant abiotic hydrogen production. You can do it, but definitely having the enzyme catalyst there really increases the chances that you're having a hydrogen issue. And so we didn't really see evidence of background hydrogen at these -400, -500 millivolt potentials.
Cool. All right. Well, let's see. I don't see, let me double check one more time, see if anyone has chimed in with any more questions, but if not.
I have one more if you have time. If not, I can email it to you, Annie.
No, sure, go for it.
I was really sort of fascinated with this lack of cell death that you observe with Shewanella, even though they weren't growing. And so, people have been trying for a long time to figure out good ways of studying maintenance energy. Are you moving in that direction? I mean, I'm assuming you've thought of this already, but having a system like that is perfect for going after that.
Yeah, well, so I will say that kind of got stricken from the paper. So I did have some paragraphs about maintenance energy, and reviewers did not like it. I think there's a lot of intense thought on maintenance energies. Yeah, I think definitely I guess one of the things that I want to be careful of is that in some ways the electrode is a little bit of an artificial environment, because it's this perfect electron donor, right? Yeah. It's just going to magically keep giving you electrons. And I think when you have oxygen there, there's also quite a bit of stress for the cells. And so they might actually need more energy under those conditions than maybe sort of your traditional maintenance energy environments, just because I think we're making, for example, oxygen-free radicals on the electrode.
Yeah. Okay. Yeah. It seems like there's something you could mess with there.
Yeah, I mean, I've talked about doing it anaerobically and potentially trying to get at something like that, but
Okay.
Shewanella is probably not a good organism to do that work in. So,
Okay.
yeah.
Cool.
But
thanks.
Yeah.
All right. Well, thank you so much, Annie. This has been awesome. So for all of you microbe nerds, we have another seminar that's coming up next week. Oh boy, let me look at my calendar and I will tell you exactly when. I think it's Tuesday, but it might be Wednesday. Let's look at the calendar. I should have been prepared for class here. But next week, we are going to be having another back-to-back micro seminar with Nick Peng from UCSB. He is a postdoc working with Dave Valentine and Michelle O'Malley, and he's going to be telling us on Tuesday next week at 11 a.m. Eastern, 8 a.m. Pacific time. He's going to be telling us about some adventurous stories of marine fungi, and I think potentially also some sort of anaerobic fungi. I can't remember exactly, but his preview is live on our website, and I'll start tweeting out some more stuff this week about Nick's seminar. So tune in this same time next week for another micro seminar to start your summer off right. All right, thanks everybody. Have a great week.